When B cells become activated by helper T cells to become effector B cells, or memory cells, which are held aside for the second infection, they essentially transform from this basic lymphocyte to a larger lymphocyte known as a plasma cell. Now, plasma cells develop an enormous rough endoplasmic reticulum. The rough ER, as you probably remember, is where protein synthesis takes place. And that allows this plasma cell, this plasma B cell, to manufacture huge quantities of these secretory receptors, receptors that are secreted to the outside of the cell and which we call antibodies. Now, what do antibodies do for us? Antibodies can do a number of things. They can cover pathogenic sites. So, for instance, in this bacterium, I gave it a pathogenic site, a dangerous site with a big mouth and sharp teeth, which of course bacteria do not have. But imagine that the antibody is acting like a muzzle on a dog. It prevents this bacterium from doing whatever it does that's so damaging. Another thing that antibodies can do is that they can agglutinate or immobilize the pathogen. So in this case, we've got a cluster of antibodies, and each one is holding on to a different bacterium, causing them to agglutinate or clump, and also making it so that these bacteria cannot spread away from one another and cause the infection to spread in the body. The third thing antibodies can do is they can precipitate things. When there are antigens or small particles that are floating free from the other pathogenic cells, these can be very difficult to clean up because they're very small. Antibodies can bind to those and precipitate them or make them visible to phagocytes. Antibodies can also opsonize. That means that they can coat or cover the outside of a pathogenic cell. That also makes it easier for phagocytes to find this cell and to engulf it. Finally, antibodies initiate complement. So here's this one yelling, "Hey, complement, come help." And complement, as you know, initiates inflammation. So all of this means that antibodies all by themselves, if they interact with a specific antigen, can trigger an immune response. And this is why they're important in our bloodstream.